Hello everyone, my name is Yuting, and I'm glad to have this opportunity to introduce our work, Evolution of Metazoan Protein Domain Toolkit, reviewed by a bird-skin model. Advances in genome sequencing are driving a growing body of work that investigates the relationship between evolution of the metazoan genome and phenotypic innovations using phylogenetic comparative method. Here, the genome is presented as a sequence of genes, represented by these colored boxes, giving a species tree and protein repertoire encoded in present-day genomes. Phylogenetic comparative methods infer the protein coding repertoire in each ancestor and the families that were gained and lost on each branch of the species tree according to an evolutionary model. Depends on the method used, different assumptions about evolutionary processes and constraints are applied. Parsimony methods assume changes are rare events and seek the reconstruction that minimizes the number of events. Dollar parsimony is a widely used parsimony method. It imposes a further constraint that a character may only be gained once but lost multiple times. Many previous studies with Dolo have reported widespread gene loss. Bird-skin models are probabilistic phylogenetic comparative methods. The evolutionary change is modeled as a stochastic process with read parameters. In this case, we seek the reconstruction that maximizes the likelihood of present-day repertoire. Bird-skin models are not widely used compared to Dolo parsimony due to the model complexity and long running times. However, BDGs make fewer constraining assumptions and output both read parameters and probability of ancestral state and state changes on the tree. In this study, we focused on what we can learn using a bird-skin model. In this work, we investigate the evolution of the protein repertoire in 21 holozoan species. In our study, the protein repertoire is represented by the protein domain families that are encoded in the genome. So what are domain families? Domains are sequence fragments that encode protein modulars with a distinct structure and function. For example, the SRC kinase shown here has 3 domain sequences that correspond to 3 separate folds. Domains are the basic building blocks of proteins and therefore a good representation of the protein repertoire. Here, we apply the BDG model to the domain family dataset. The BDG model inferred domain gain, loss, duplication rates, and the expected number and size of domain families in each ancestral node, also the expected gain, loss, expansion, and contraction of families on each branch. With the domain rates, we first look into the relationship between domain event rates and domain function using a manually curated domain-specific ontology, in which each domain is assigned to one of the 50 functional categories. This figure shows the distributions of gain, loss, and duplication rates in each functional category. Functions are ordered by the median duplication rate, with slow rates on the left and fast rates on the right. We can see that domains associated with basic cellular functions, such as translation, RNA processing, transcriptions, tend to have slow rates, where domains with functions related to metazoan innovations such as immune response, cell adhesion, signal transductions tend to have fast rates. To assess the plasticity of the ancestral repertoire, we used the bird-skin to infer the expected number family gains and losses on each branch, as shown in the first column of bar plot. Each bar shows the family gained, the families lost, and the net change. Similarly, we inferred for each branch the expected number family expansion and contraction, as shown in the second column of bar plots. Overall, we see bird-skin infers a history dominated by remodeling and turnovers, and only few branches show streamlining. This is very different from the widespread loss reported from previous studies using Dolo parsimony. As we mentioned earlier, birthday skin is a more flexible model that can explain a patchy distribution with parallel gains and parallel losses. On the other hand, Dolo can only explain a patchy distribution with parallel losses. As an example, here shows one family with a patchy distribution. For this family, birthday skin would infer 2 gains and 1 loss event. However, Dolo parsimony would infer 1 gain and 12 losses. Naturally, we wonder if the assumptions of Dolo parsimony contribute to reports of widespread losses. So we applied Dolo to the same dataset for testing. On average, birthday skin inferred 1.7 losses per gain, while Dolo parsimony inferred 7.5 losses per gain, which is almost 5 times larger than that of birthday skin. So our results show that Dolo clearly has an overwhelming preference for family losses compared with birthday skin model. In conclusion, we found that the evolutionary rates inferred by birthday skin models are a promising source of biological insights. Reconstruction with birthday skin suggests an ongoing evolutionary remodeling of the protein repertoire, while reconstruction of Dolo parsimony results in almost 5-fold increases in the ratio of family losses to family gains. This indicates that model choice can dramatically impact final conclusion. We also wonder whether the perceived importance of genome reduction as an evolutionary mode might be an artifact of the model used. My colleague, Dr. Marie Stozer, will give an in-person talk. Our paper is also available on bioRxiv. Please contact us if you have questions, and thank you for your time.